Hello, everybody. My name is Leonardo DallAgnol. I'm a professor at the Federal University of Maranhão in Brazil. So today I'll be presenting our work that is entitled Assembling, Characterization, and Genome Prospecting from Enriched Metagenomes of 3 cyanobacteria from Cerrado biome in Brazil. This is a collective work that was conducted in the LabGen and GB3 work group, where I'm one of the responsibles, but also we have a lot of students and other professors from different institutions in Brazil. So I will be going forward now. Okay. So this work was done in the Chapada das Mesas National Park. It's a park located in the state of Maranhão. It's a state in the northeast of Brazil, yeah, and the national park is located here in the south of the state. It's a bit far away, so it's around 700 kilometers from the capital where we work. And we did part of this project that was a microbiome metagenome analysis of the sediments on the park. So we collected some samples in the Farinha rivers and also in some springs outside of the park. So from these samples of sediments and water, we were able to isolate some cyanobacteria. So here we have some of the samples we isolated, non-axenic culture. So we got Geminocystis lineage, also Nostoc strain and Pantanalinema strain. These two, both of them were pictures we're taken at our group. And speaking about the strains we have, the Pantanalinema was initially described in saline lake waters in Pantanal, that's for the name, in Brazil biome. And this was the first time we located this outside of Pantanal in Cerrado biome. And also our work was the first genome of the genome. So it was very interesting for us to do this work. Also, the Geminocystis has an important ecological role. It's previously identified as Geminocystis and has a very little information about this genome content and physiological properties. And Nostoc species belong to the family Nostocaceae and is a rich source of active compounds. So it's very interesting to look about this potential biotechnology compounds that it has on its genomes. The goals of the work was to improve genome annotation, especially hypothetical proteins, and to do genome mining on the isolation. So for gene sequencing, we used MySeq reagent kit 2 from Illumina, and for the assembly, we did Metawrap using metaSPAdes, and for the minor model, we use CONCOCT and metaSPAdes 2. And then for annotation, we use RAST and prokaryote genome annotation pipeline. We did AntSmash, CRISPR-Cas finder, FASTERS, and for the phylogenomics, we did GS and GGDC. So for the hypothetical proteins, we annotate physical-chemical parameters, sub-cellular locations, and also domain of family using these servers and softwares here. So we find a lot of interesting functional categories. For example, for Nostoc, we found 25 categories, functional categories, also for Pantanalinema. And the phylogenomics analysis confirmed the morphological identification we did. So for the genome mining, we found 14, and 24 respectively from Geminocystis, Pantanalinema, and Nostoc. And as I said, Nostoc it's well known for being a prolific producer of secondary metabolites. So we can see here it has a lot of important biosynthetic gene clusters, for example, for microviridines, from microcysteines, and a lot of NRPS and PKS biosynthetic gene cluster types. And this microcysteine is hepatotoxins, and this is important, for example, for us to see because the national park, it's a very touristic area, so there's a lot of touristic tourist influx. So it's important to see if we have any toxigenic cyanobacteria in the area. Despite that never happened any accident in the park, it's interesting to know that they are there and to improve the monitoring protocols. So for the hypothetical proteins, it's interesting to see that more or less half of the CDS annotated in the genomes usually were hypothetical proteins, as they are environmental samples, they're environmental strains. We don't have much information about the genomic potential. So a lot of annotation that is done automatic, it doesn't find really what they do or what the potential they have. So we started doing trying to improve the annotation using these servers like Smart, Interpro, Catch, PIFA, and CDD. And we compare all of them for each genome. So we can see that usually the Interpro was the one that have the better results. Also Smart was the second or the first one. And PIFA and CAT was the worst for annotation, for improving the annotation of hypothetical protein. So about the cellular location, we found that usually most of them that was expected was cytoplasmatic, a bit of them on membrane, and a bit of them extracellular. And in some cases, it was unknown. 20% in Nostoc and 21% in Pantanalinema. And in Geminocystis, we didn't find anyone know because it's compared to Synecocystis, well-known genera, we could find the location of all hypothetical proteins. About the phylogenomics, we were able to place them. For example, in Geminocystis, it was placed close to Geminocystis genera, and about Pantanalinema, it confirmed that it was in the Leptolimbia family. It doesn't have another genome to compare in the genera, so it was the Leptolimbia was the closest we got. And for Nostoc, it also confirmed it was very close to Nostoc Linkia, but it's not close enough by the DNA hybridization to say it's the same genera. So the analysis revealed a lot of interesting genes and biotechnological potential. It's interesting to find some toxin genes, so it's important to improve the monitoring in the park and in the springs outside. And we can find a great potential of producing bioactive compounds and secondary metabolites. So it's a way to improve the value of biodiversity in the biome like Cerrado, that it's very under stress now in Brazil. And I would like to thank all the State Foundation, Science Foundation, CAPS, and PK that funded the research and all the partners in this work. Okay, thank you.